Good morning, everyone. My name is Jiwon Kim. I am studying at Professor Inwon Kim's laboratory in Korea University. I am so glad to make a presentation about my research today. The title of my research is Lipase-catalyzed preparation of Neopentyl Glycol Diester as a biolubricant. I'll be presenting as following order: introduction, objective, procedure, results, and conclusion. The first is about introduction and objective. Before I explain Neopentyl Glycol Diester in detail, I would like to briefly talk about lubricants. What are lubricants? The comprehensive definition of lubricants is substances used to reduce friction between surfaces, and they are defined as various compounds like fluids, oils, and greases. However, in addition to industrial applications, lubricants are used for many other purposes. As you can see, there are various functions of lubricants. Then, what are lubricants made of? Typically, lubricants consist of 90% of base oil and 10% of additives. There are four types of lubricants that can be classified depending on this base oil, which is categorized as mineral oil and bio oil. Mineral oil is sourced from petroleum crude, and bio oil is sourced from natural oil. Some oils are structurally modified by chemical reaction for increasing some properties and performances, and these are all called synthetic oils. On the right side of this slide, there is a graph about the cost and performance of lubricant-based oils. Until the second half of the 19th century, nearly all lubricants were based on natural vegetable oil or animal oils and fats. After the Industrial Revolution, mineral oil lubricants began to be used a lot. However, the recent issue of lubricant has a lot to do with the environment. Mineral oil-based lubricants are not biodegradable and environmentally toxic. So, biolubricants began to be used more to solve these problems. Biolubricant is the term used throughout the lubricant using natural renewable resources like vegetable oils. It is biodegradable and non-toxic. Therefore, interest in biolubricants has recently grown due to the environmental issue. Moreover, bio-based synthetic oils are a good compromise suggestion considering good performance, cost, and environment. So, we studied one of biolubricants, Neopentyl Glycol Diester. It is synthesized through the reaction of neopentyl glycol with fatty acids from vegetable oils. It can be used as hydraulic fluid on the industry, and mostly it has been produced using chemical catalysts. The aim of this study is to synthesize neopentyl glycol diester using an immobilized lipase. The next is procedure of our study. This is the reaction scheme of MPG diester synthesis. 1 mole of neopentyl glycol reacted with 2 mole of fatty acid and produced 1 mole of MPG diester. In this study, we use immobilized lipase as a biocatalyst of esterification. So, before the esterification reaction, immobilization of enzyme was preceded. For enzyme immobilization, we prepared with Eversa transform 2.0 lipase. which is developed for biodiesel production. It is liquid-type enzyme and relatively cheap than other commercial lipases. As a carrier, Lewatit VP OC 1600 was used. It is hydrophobic and macroporous. This diagram shows the general method of enzyme immobilization in our lab. First, enzyme solution was prepared with addition of sodium phosphate buffer in Eversa transform 2.0, which is liquid lipase. At the same time, a hydrophobic carrier Lubricant needed to pre-soak in ethanol to allow the enzyme to physically enter the pores of the carrier. Next, the mixture of enzyme solution and carrier was added to a flask, and it was placed on a water bath shaker for 17 hours. Then, it was washed with buffer solution. Finally, we can get immobilized lipase. Enzymes are vulnerable to water, so before reaction, we pre-equilibrate enzyme using saturated salt solution water activity chamber. The method is simple. Fill the sealed container with about a quarter of the saturated salt solution. After that, a net is placed on top of it, and the enzyme is stored for about 4 days. When the immobilized lipase with desirable water activity was prepared, the esterification will begin. This diagram shows batch reaction system I used for MPG diester synthesis. The esterification of MPG and fatty acid was performed in the flat-bottom glass vessel equipped with water jacket for temperature control. To remove water, water vacuum pump was installed and connected to top of the glass reactor. The reaction was initiated with adding the enzyme. From now on, I am going to talk about the results. First, for enzyme screening. 3 commercial lipase and immobilized lipase prepared in our lab were tested to decide the best enzyme for the reaction. The highest activity was achieved with our immobilized lipase. Our immobilized lipase showed much faster rate and higher conversion during the entire reaction compared with Novozyme 435, which was reported as best enzyme for synthesis of MPG diester. Second, for the enzyme loading. It is an important factor because of economic viability, thus must be used as small as possible. Conversion increased significantly when enzyme loading was increased from 1 to 5%. However, there are no significant differences in the conversion between 5 and 7.5%. Therefore, we selected 5% enzyme loading for the next parameter experiment. Third, for the temperature. Temperature is one of the crucial factors because of enzyme activity, residual activity, energy saving, etc. The conversion obtained at temperatures of 50 and 60 degrees Celsius increased significantly with time during the first 6 hours and then approached equilibrium at longer time. So, 50 degrees Celsius was chosen as optimum temperature. Fifth, for the vacuum level. To remove water for preventing undesired reaction like hydrolysis, vacuum system was conducted in this reaction. Maximum conversion of 63% was obtained under no vacuum at 10 hours. However, When we applied vacuum, the conversion increased markedly up to 97% at 8 hours, and there was no significant difference in the conversion between 10 and 200 torr. Therefore, we selected 200 torr as an optimum vacuum considering energy saving. Maximum 97% of conversion was achieved by applying vacuum. However, vacuum needs energy, so considering economic feasibility and maximum conversion, we came up a new experimental strategy called vacuum combination method. For vacuum combination method, 6 different vacuum protocols were employed. Control protocol is reaction at a constant vacuum of 200 torr for 10 hours, and the combination protocols for the reaction occurred at no vacuum for first certain hour, followed by reaction at vacuum of 200 torr for an additional hour. The total reaction time is 10 hours. From these trials, we demonstrated that there was no significant difference in the conversions obtained between constant vacuum and combination of vacuum and no vacuum for 1 or 2 hours of entire reaction time after 10 hours. Therefore, as a novel strategy, the effects of combination of vacuum and no vacuum on the synthesis of MPG diester were investigated. This is the final part conclusion. In conclusion, MPG diester was successfully synthesized via immobilized enzyme catalyzed esterification. Finally, these are the highlights of our research. We used immobilized lipase and it showed extraordinary activity for synthesis of MPG diester. With low vacuum as 200 torr, the conversion increased markedly up to 97% from 67%. And we proposed a novel strategy, the vacuum combination method. Thank you for listening.